students, coming to the next class is the chemical composition of the cells. In the last class we discussed about introduction for the biomolecule chapter. In that case we discussed about that basic elements and the molecules. See, a group of carbon, hydrogen, oxygen, nitrogen, phosphorus, these compounds combines produces that molecules, right? Here again the molecules combine produces the compounds. See, there is a difference is there between the living and the non-living cells. Within living cells there is a compounds combined produces cell organelles, right? That cell organelles combined produces cells, and the cells are combined produces tissues. The tissues are combined produces organs. Organs are combined produces organ system. A group of organ systems are combined produces the organism. So that is about that living things, and coming to the non-living things, there is no formation of cell organelles. So here what I am telling, whatever that compounds are there, the best example, sodium chloride not have the life, it's unable to form the cell organelles. See, water also not have the life. So here what I am telling, a group of molecules are combined which produces liquids and the compounds which are non-living things, right? So non-living things, the cell organelle state is absent. In living cells the cell organelle state is present. So that is about that living and non-living organisms differentiation we have seen in the last class, we discussed, right? Coming to this class, today we are going to discuss about that chemical composition of the cells. There is a different types of cells are there. See, the cells, the chemical composition also different. Plant cell chemical composition is different, and different from animal cells. Animal cells chemical composition is different from the bacteria cells. So, but the basic compounds are similar. Basic compounds means here proteins, lipids, carbohydrates, nucleic acids. These are common in all the cells. These are common in all the cells. So, amino acids, right, and some of the acetyl-CoA molecules and other components are common, but the ratio is different. Maybe in plant cell, okay, one type of proteins are more. In animal cell, another type of proteins are more. So the ratio is different, but basically all these cells are having carbohydrates, lipids, nucleic acids, proteins. So we are here discussing about that chemical composition of cells. In that case, there is two methods are there. One is that living cell method, other one is that dry cell method. See, in one case, in one method we are using the living cells, in another method we are using the dry cells. See, both the methods are different pathways. Both the methods are different pathways. See, in living cells, okay, we are estimating one type of molecules. In dry cells, okay, when we are using the dry cells, the method is different. So when we are using that living cells, first of all here we are discussing about that living tissues, right? In that case, this is the one of the living tissue method. Sir, what kind of living tissue we are collected here? Simple, liver tissue. We are eating chicken, right? We are eating that mutton. In that case, if we went to that slaughterhouse, that time we can collect that liver, right? Maybe the chicken liver, maybe the goat liver, right, or sheep liver we can collect. That livers, okay, that liver cells are subjecting for that tissue analysis process, not tissue analysis exactly here, chemical composition analysis process, chemical composition analysis process. So here we are using one of that pistil and the mortar. We are using that pistil and the mortar. See, most of the students we are seeing so many times. I thought I'm expecting Miss Walk, one of the toothpaste ad, right? Within that ad, one of the person, okay, grinding, right? So there is a bowl is there and there is a holder is there, right? The bowl and the holder, both these are using for the grinding purpose. See, these are nothing but pistil and the mortar. See, the bowl is called as pistil, whatever that holder is there, that one is called as mortar. So here, this is the pistil and this is the mortar. So the pistil and mortar using for the homogenization process. Homogenization is nothing but grinding of tissue. Grinding of tissue. So how we are doing that grinding process or homogenization process? Very simple. Whatever that liver tissue we are collected, whatever that plant leaf we are collected, or maybe we are collected one of the vegetable, we are collected any one of the tissue or the leaf or any vegetable, right? That particular part chopped into that small pieces, right? All these pieces are added into that pistil. Then we are applying the mechanical pressure. We are applying the mechanical pressure. So if we apply the mechanical pressure by the usage of the motor, whatever that tissue contains cells are there, the cells are ruptured. See, in that case we are adding one of the chemical reagent also. Name of the reagent is trichloroacetic acid. Trichloroacetic acid. All of you remember, whatever that chopped material is there, we are added into the bubble. After that addition process, we are adding one of the chemical reagent. The reagent name is the trichloroacetic acid. So presence of trichloroacetic acid, we are doing that complete grinding process. We are doing the complete grinding process. So presence of the chemical substance, here completely the cells are destructed. Means cells, leaf is the tissue. The tissue, we know, group of cells. The cells are destructed, right? Maybe the liver tissue. Liver is the tissue, right? Liver tissue, right? The cells are, if we apply the mechanical pressure, presence of the trichloroacetic acid, the cells are destructed, right? So after that destruction process, we are subjecting for the filtration. The step is called as filtration. So how we are doing that filtration process? Suppose this is the one of the tissue, right? This is the tissue. We are using here the cheesecloth. We are using here cheesecloth. This is the one of the cheesecloth, right? So we are using cheesecloth. By the usage of cheesecloth, remember we are adding, okay, whatever that grinded material is there, or whatever that sap, after the grinding process, what we get, that liquid like substance, that one is called as sap, right? The sap is subjecting for the filtration process by the usage of cheesecloth. So by the usage of cheesecloth, we are doing the filtration process. Okay, after that filtration process within the tissue, we have two zones here. We have the two zones. So here, okay, presence of trichloroacetic acid, grinding process. After completion of grinding, then we are subjecting for the filtration, right? Filtration done by the cheesecloth. Right? After that filtration process within the tissue, we got two zones here. See, the bottom side zone is called as fillet. Upper side, there is a supernatant is present. So after the filtration process within the tissue, we got two zones. One of that bottom side zone is called as fillet. Upper side zone is called as supernatant, right? So these zones, okay, another names also given. For these two zones, another names also given. See, upper side zone is called as acid soluble region, right? See, lower side zone is called as acid insoluble region. So why here added acid? Remember, we are using here trichloroacetic acid. So after that filtration process, the acid is present. In the both the zones. Then other one is that soluble region. Okay, acid soluble region means whatever the components which are present within that soluble region, all these are soluble components or molecules which are present upper side region, which are soluble. That's the reason why the upper side region is called as acid soluble region, right? And coming to the lower side, whatever the components are present, okay, which are insoluble, right? That's the reason why this region is called as acid insoluble region. So one of the region is called as acid soluble region, other region is called as acid insoluble region. Then here molecules are separated. See, in one case, acid soluble region contain components, other case, acid insoluble region contain components. One is acid soluble region contain components, other one is acid insoluble region contains components. So here acid soluble region, there is in Macromolecules. Whatever the components which are present, in soluble region, whatever the components are present in soluble region. See, these are called as biomicromolecules. These are called as biomicromolecules. Why these are called as biomicromolecules? Because these are having small size and the molecule size, size is small, right? The molecular weight is less than 1000 Daltons. The molecular weight is less than 1000 Daltons. So according to the biochemistry, any molecule which are present in the living cells, which are having less than 1000 Daltons, which are called as biomicromolecules. Remember, this is biochemistry. Okay, the components which are present in the cells which are having less than 1000 Daltons, that molecules are considered as biomicromolecules, right? So here biomicromolecules examples are amino acids, monosaccharides, acetyl-CoA, nitrogen bases. Remember, we already discussed amino acids which are supporting for the formation or the synthesis of proteins. A group of amino acids are combined by the peptide bond which are forms the proteins, right? Monosaccharides, glucose is the monosaccharide, fructose is the monosaccharide. So a group of monosaccharides are combined produces the polysaccharides. So here we are talking about the individual structures. Not the group structures, right? Not the condensed structures. And the acetyl-CoA, we know about the acetyl-CoA, that one in the last class also we discussed. A group of acetyl-CoA molecules are involved in the fatty acid synthesis, right? And nitrogen bases, within the DNA or within the RNA there is an adenine, guanine, thymine, cytosine, right? And uracil is present in the case of the RNA, right? So these are the nitrogen bases. Nitrogen bases are present. So, sir, why these are soluble? Why all these components are soluble? Remember, these are having very small size. The molecular weight also less, and the molecular weight also low. So presence of low molecular weight, presence of very small size, easily soluble. Which are easily soluble, right? So that is about the acid soluble region contains components. Right? That is about the acid soluble region. This is the upper side. The blue color region indicates here acid soluble region. Other side, acid insoluble region. All of you remember here, insoluble means the components are not soluble in the acid. Which are insoluble. So those are the compounds which are present in the lower side, which are not, the components are not soluble. That's the reason why this region is called as acid insoluble region, right? And which are biomacromolecules. All of you remember, these are micromolecules. Micro means small. Biomicromolecules. Bio indicates like is nothing but here cells. Biomicro means small size. Biomacro means large size, right? Biomacromolecules. So here biomacromolecules are having more than 1000 Daltons. Biomacromolecules are having more than 1000 Daltons, right? Here examples are proteins are the best examples, right? And nucleic acids are the best examples, and polysaccharides are the best example. I will discuss definitely lipids also there, but lipid case is quite different. I will discuss later regarding to the lipids. Like proteins. See, proteins are the true biomacromolecules. Proteins, nucleic acids, polysaccharides. All these are true biomacromolecules or biomacromolecules. Why these are considered as biomacromolecules means which are having more than 1000 Daltons. The molecular weight is more than 1000 Daltons. How proteins are formed? A group of biomicromolecules means here a group of amino acids are combined, produces proteins. A group of monosaccharides are combined, produces polysaccharides. Okay. A group of nitrogen bases, ribose sugar or deoxyribose sugar, phosphate, which are combined, produces the nucleic acids. So that is about that formation of macromolecules. Biomacromolecules. So biomacromolecules which are having large size, the size is large, molecular weight is high. High means here more than 1000 daltons. Right? These are considered as biomacromolecules. Then I have to discuss about the lipids. Lipids are quite different one. Lipids, these are not considered as true. These are not considered as true bio micro. These are not considered. Sorry. Macro. Yeah. Lipids are not considered as true bio macro molecules. Remember, these are macro molecules, right? So why lipids are not considered as true bio macro molecules? Remember, which are having 800 daltons. The molecular weight is 800 daltons. As per the statement, biomacromolecules, all biomacromolecules are having more than 1000 daltons. If less than 1000 daltons, which are considered as micromolecules, macromolecules having more than 1000 daltons, micromolecules are having less than 1000 daltons. See, lipids are having around 800 daltons. So as per this statement, which are definitely considered as biomicromolecules, but which are included in biomacromolecules, So why these are considered as biomacromolecules, which are present in insoluble region, All of you remember, generally we know lipids are one of that organic substances, right, which are insoluble in water, soluble in organic solvents. Lipids are organic substances which are insoluble in water, soluble in organic substances. So here lipids are present in insoluble. Lipids are present in insoluble region. Lipids are present in the insoluble region. Means lipids are not soluble here. And see, lipids are not soluble, right? And next one, lipids are having high. Large size. Lipids are having large size. And lipids are having large size. And lipids are having low density. Remember, especially the lipids are insoluble. So lipids are insoluble, which are having large size, which are having low density. So here what I am telling, whatever that lipid components are there, the lipid components are not soluble. Lipid components are insoluble, which are having 800 daltons molecular weight. Even though these are considered as biomacromolecules, because which are present in insoluble fraction. That is the one of the reason. And next one is which are having large size, but density is low. The density is the low density. That is the reason why here lipids are considered as biomacromolecules, but these are not true biomacromolecules. Lipids are considered as biomacromolecules, but these are not true biomacromolecules. This is the one of the bit given in the NEET examination so many number of times. Lipids are not strict biomacromolecules, because which are present in insoluble fraction, which are having large size, and which are having low density. Option is fourth option, all of the above, right? So that is about that lipids, right? So that is about that insoluble region and the soluble region. So then we we discussed about here majorly two types of components. Yeah, regarding to the two types of components. One is bio micro molecules, other one is bio macro molecules. Whatever the tissue contains that cells are there. Okay, bio micro molecules, other one is the bio macro molecules. So here less than 1000 atoms, here more than 1000 atoms. Simple, amino acids, monosaccharides, acetyl-CoA, nitrogen bases. Here proteins, nucleic acids, polysaccharides. The last one is the lipids. Lipids are exceptional cases. And next one we will discuss about the as analysis method. As analysis method, here we are collecting the dry tissue. Here we collected the living tissue. The basic difference is the living and the dry. See, the living cells contains that sufficient amount of the water. Dry cells means almost is a dead. Whatever the tissue contains, water is there, the water is evaporated. So here we are collected the dry tissue. Right? Here we are collected the living tissue. Right? We'll discuss about that ash analysis method. Next one. Coming to the next one is the ash analysis method. In that case, we are already collected the dry tissue. The dry tissue is subjecting for the ash analysis method. Here we are having that very special small bowel-like structures or small bowels, right, which are made up of with the silica, right, which are made up of with the silica, which are called as silica crucibles. Special small bowel-like structures which are called as silica crucibles. See, whatever that dry tissue we are collected, we are chopped and we are added. We are chopped the dry tissue, we are added to inside the silica crucibles. Inside the silica crucibles. Maybe the dry tissue after the complete drying process, maybe in certain cases that one is modified into the powder structure. After the complete drying process, that one is subjecting for that flour. I see we know about the different types of flours, wheat flour and different flours we have, right? So here also same thing, whatever the dry tissue we are collected, that one is subjecting for that complete breakdown process. Finally we get that flour, right? So that flour also added to that silica crucibles, right? That silica crucibles kept inside the muffle muffle furnace. So here one of that device is there. The device is called as muffle furnace, right? Muffle furnace, right? Inside that muffle furnace we kept that silica crucibles. The silica crucibles already having that material or the dry tissue, right? Inside that inside the muffle furnace we maintain up to the 1000 degrees centigrade. We maintain up to the 1000 degrees centigrade. At 1000 degrees centigrade, whatever that carbon contained components are there, which are released in the form of carbon dioxide. See, completely water is evaporated. The carbon compounds are evaporated. Evaporation means here the carbon compounds are converted into carbon dioxide. The water content is changed into the water vapor. So both these are eliminated. So in a cell, whatever that amino acids are there, whatever that remaining all components are there, see the carbon contents are majorly converted into the carbon dioxide. Then presence of 100 degrees also the water forms that water vapor. So at 1000 degrees centigrade, see whatever that molecules are there, all these molecules are completely degraded. Finally produces the ash. Remember why we are collecting here ash. Finally we are collecting here ash. Why we are collecting ash? Remember the ash collection is majorly supporting for the estimation of different types of minerals which are present in the cell. For suppose within the bone cells we are having the more amount of calcium, and within that different types of cells are there, different types of mineral composition is there. So what I am telling here, sir, how we are estimating the minerals in a cell? How we are estimating the minerals in a cell? By the ash method. By the ash analysis method, we are estimating the different types of minerals, right? So here the ash is present, right? The ash is collected. That one is subjecting for the titration methods. By the titration methods, we are analyzing the different types of minerals, or we are estimating the different types of minerals. So very simple. Here one of the device is there, that one is called as muffle furnace. Here this one is maintains up to 1000 degrees centigrade, more than 1000 degrees centigrade also sometimes, right? So here silica crucibles we have, which are having the heat resistance, which are also stable at 1000 degrees centigrade, right? Then whatever that fluor content is there, the fluor content is added to that fluor content or the dried material added to the silica crucibles or added inside the silica crucibles. Finally we get then ash. The ash is subjecting for the titration method. After the titration process we are estimating the different types of minerals, right? So based on that, in a cell what type of minerals are present we are estimating. So this is the ash analysis method. So based on that living tissue, okay, or pistil and mortar method, pistil and mortar method, or the living tissue collection method, we are estimating that biomicromolecules and biomacromolecules. Okay, based on that ash analysis method, we are estimating the different types of minerals inside the cell or in a cell, right? That is about that the composition of the cells. Composition of the cells. Next one we will discuss about that another topic, right?